So our first presenter today will be Tom Givnish, a plant ecologist and evolutionary biologist, and the Henry Ellen Gleason Professor of Botany and Environmental Studies at the University of Wisconsin-Madison. Tom and his students have made seminal contributions to our understanding of plant adaptations for energy capture and reproduction, and the study of adaptive radiation, as well as determinants of plant species diversity using a variety of methods, including phylogeny, ecology, geography, and a number of others. His interests are broad-ranging and include orchids, thankfully, and he's especially interested in adaptive radiation of plants. And today, he will start us off by talking about the drivers of biodiversity. So with that, I will stop sharing. And Tom, whenever you're ready, please go ahead and share your screen.
Okay. Hi, everyone. Just take me a few moments here to get connected properly and to show my presentation. Let me just see here. I've got to go to setting this up. Can you see that, everyone?
Yep.
All right.
Yeah, looks
Anyway,
good.
good morning, everyone. Today I'd like to talk about some recent advances in our understanding of relationships among orchids and what they tell us about the causes of their extraordinary species diversity based on variation in orchid morphology and ecology and their historic pattern of geographic spread across the globe. These insights in turn may suggest some additional directions and strategies for global orchid conservation. Now, orchids are one of the crown jewels of planetary biodiversity. Orchidaceae is the largest family of flowering plants with over 750 genera and 29,000 species. They are more diverse than birds, mammals, and reptiles combined and grow in a wide range of habitats on every continent except Antarctica. Essentially, all temperate orchids are terrestrial ground dwellers, but most orchids inhabit tropical forests, and over 80% of those are epiphytic and adapted for growth on tree trunks, branches, and twigs. Orchids display extraordinary floral diversity and sexual selection run rampant, with species showing remarkable adaptations to individual pollinators, and with different orchid species often using the same pollinator without hybridizing via precise placement of pollen packets or pollinia on different parts of that pollinator's body. Now, plant biologists, ecologists, and evolutionary biologists have long puzzled over why orchids are so diverse and how their diversity is related to their defining morphological traits, the wide range of habitats they occupy, their reproductive biology, their interactions with other species, and their history of geographic spread across the planet. Today, I will attempt to address each of these issues based on studies by many orchid biologists over the past several decades and on research that my colleagues and I have conducted over the past several years. Now, the family Orchidaceae is one of 77 families of monocots, all of which bear a single cotyledon or seed leaf and usually bear adult leaves with parallel venation and flowers with 3 petals, 3 sepals, 3 stamens, and 1 to 3 pistils. Based on variation in the DNA sequences of 4 genes from the chloroplast genome, Mark Chase and his colleagues in 2006 produced a family tree or phylogeny of the monocots and showed that orchids are members of one of the 12 orders of monocots, namely the order Asparagales. Now, my colleagues and I recently extended this analysis by including 5 times as many species, including nearly all the families of monocots, and 12 times as much sequence data per sample. Adding many more species and much more data per species has greatly strengthened support throughout the tree. That's the reliability of our inferences of relationships. And as you can see, the brightly colored branches in the Chase et al. Analysis, which indicated low statistical support, have largely disappeared. The blue dots on top indicate families whose position have changed between the 2 analyses, and the red dots indicate families that we added to the analysis. Now, as you can see, the Orchidaceae is sister to the lineage formed by the 13 other families of order Asparagales, which include those housing the agaves and yuccas and hostas, crocuses, aloes, amaryllids, and irises, as well as several relatively small groups from the southern hemisphere. The order Asparagales itself is the closest relative of a group of 5 closely related orders known as the commelinids, which are highlighted here in that red box. Now, orchids differ from all other monocots, and indeed all other flowering plants, in possessing a combination of 4 defining morphological characteristics. A labellum, a large, specialized, often colorful lower petal that serves as a landing platform or an attractant for pollinators. A column on which both the male anthers and a single female stigma are borne in the center of a bilaterally symmetric flower. Tiny seeds, each composed of an embryo of just a few cells and lacking endosperm in which other plants store food reserves. And mycoheterotrophic germination, in which fungal infection is required for orchid seeds to begin development, during which the seedlings depend entirely on fungi, not photosynthesis, for energy in the form of sugars. Tiny seeds and mycoheterotrophic germination are almost surely co-adapted, in that small seed size allows a plant to produce and disperse huge numbers of wind-dispersed seeds over long distances. Vast number of seeds should allow at least some to find just the right fungal partner. Conversely, energetic input from fungal mutualists may be required to produce seedlings from just a few initial cells. Tiny seeds can also be seen as a pre-adaptation for epiphytism, which requires the ability of seeds to land and remain on narrow twigs or branches or vertical tree trunks. Now, what distinguishes orchids from other plants ecologically? Orchids are remarkable for having large numbers of species that are epiphytic, more than any other family, indeed, of plants. And for large numbers of the species growing in the tropics, again, more than any other family of plants. They also are notable for having exceptionally strong mycorrhizal associations with mutualistic fungi, and for displaying an extraordinary diversity of adaptations for pollination, including extreme specialization on individual pollinators and the widespread use of deceit in attracting pollinators without any material reward. Of course, those adaptations apply to individual species, but large numbers of them. Fully mycoheterotrophic plants have also evolved more than 30 times in the orchids, far more frequently than in any other plant group. These so-called holomycoheterotrophs have gone over completely to the dark side, obtaining energy entirely from fungi, not photosynthesis. The great frequency of so-called independent origins of this strategy most likely reflects the shared fact that all orchids are mycoheterotrophic early in life, and thus have the genetic components on board to feed on fungi and to do without developing chlorophyll or leaves or most roots. Such full mycoheterotrophy is unlikely, however, to have been a major driver of orchid diversification, given that fewer than 300 species have evolved this physiological strategy. Orchids are best known for displaying an extraordinary range of adaptations for pollination. Widespread interest in this topic traces back to Charles Darwin, who published a landmark book on orchid pollination in 1862, just 3 years after On the Origin of Species appeared. After Darwin moved to Down House outside London in 1841, he and his family frequently walked in the surrounding area. He was especially fond of a location, which still exists today, that he called Orchis Bank, in which many native orchids grow in profusion under the trees or over chalk limestone in open grassy areas. Darwin had read Christian Sprengel's German book on the natural fertilization of flowers, and it set him to thinking about how the strange structure of many orchid flowers might represent adaptations to attract animals to move pollen from plant to plant. What began, as a recreation became a mania, as Darwin corresponded far and wide with gardeners and botanists seeking observations on native and exotic orchids. As his enthusiasm mounted, he wrote to his close friend, Joseph Hooker, then director of Kew Gardens, that, quote, "you cannot conceive how the orchids have delighted me," that, quote, "the orchids have been splendid sport," and that, quote, "the contrivances of orchids beat, I think, any animal." And then he wrote to orchid expert John Linley that, quote, "orchids have interested me more than almost anything in my life." Coming from Darwin, that is high praise indeed. Now, through observations and experiments, Darwin showed that bees visiting orchid flowers in Great Britain land on the labellum and become precisely aligned with the pollen masses, or pollinia, and the stigma on the floral column. Pollinia each carry enough pollen grains to fertilize thousands to millions of seeds produced by a single flower. Then, more remarkably, the sticky feet of the pollinia become glued to the backs of bees, or pencils, as the bees force their way into the nectar-bearing gullet of flowers and then pull out. Several minutes have to elapse for the pollinia to dry and to twist into shape, so that when an insect or pencil returns to another flower of the same species, the pollinia can then and only then contact the stigma. This quote unquote contrivance guarantees that a plant will not self if the time required to twist exceeds the time a pollinator spends visiting flowers on a given plant. Such a delay would allow a plant to gain all the advantages that accrue from outcrossing. The pollinators, of course, benefit from, in this case at least, sipping nectar while visiting the orchid flowers. Recently, Peter and Johnson confirmed Darwin's outcrossing idea by showing that across over a dozen species of orchids, the time required for the pollinia to twist and become capable of fertilization did in fact exceed the visitation time. Now, bees, moths, and butterflies were among the pollinators Darwin observed, with pollinia glued repeatedly and precisely to specific points on their backs or proboscis or eyes, guaranteeing that after the pollinia twist, they're in the right position and can thus fertilize the orchid in question. These studies laid the foundation for studying coevolution between different groups of organisms, which remains a hot area of research today, and can involve, for example, the reciprocal selection pressures and reciprocal evolution involving plants and their pollinators, plants and their herbivores, or more generally, say, hosts and their pathogens, or predators and their prey. Darwin found that orchids pollinated by moths and butterflies have long nectar spurs that fit the unfurled length of the proboscis of their pollinator. He argued that competition among orchids for pollinators would select for greater nectar rewards and thus longer spurs, and result in the evolution of moths and butterflies with longer and longer proboscis. Based on the discovery on Madagascar of a remarkable orchid, Angricum sesquipedale, with a nectar spur up to 18 inches in length, pale flowers, and a strong scent emitted at night, Darwin predicted that someone someday would find a gigantic moth with a proboscis more than a foot long that pollinated. This prediction seemed outlandish at the time, but just such a moth was found in 1903, although it was only demonstrated to actually pollinate the plant in 2011. In recent years, two closely related species of Angricum with much smaller flowers and highly reduced spurs, both of them native to the small island of La Réunion, 500 miles east of Madagascar in the Indian Ocean. So quite a isolated part of the world where few pollinators would ever have arrived via dispersal. And it's shown that it has been shown that these species are pollinated in one case by small nectar-eating birds known as white-eyes, as you see here. And another species with even smaller flowers is pollinated by crickets, which is a global first. Yet another close relative of Darwin's orchid, the ghost orchid from South Florida, the famous star of the orchid thief, is pollinated by the fig hawk moth and related species, all with long proboscis. Now, orchids can provide rewards other than nectar. We now know that many South American orchids are pollinated by male euglossine bees, which are drawn to their flowers to gather fragrances they stuff into pockets on their legs and make into perfumes that attract their own mates. Small chemical changes in such fragrances might attract different euglossine bees and lead promptly to reproductive isolation and rapid speciation in such orchids. Now, Darwin did miss two really big things about orchid pollination. First, ironically, given his interest in speciation and in pollination, he did not understand that species in several orchid lineages have rapidly evolved flowers adapted to different pollinators. Presumably, such an adaptive radiation is tied not only to ecological specialization, but also reproductive isolation, and has led to a multiplication of species. The first demonstration of this came from my student Jeff Hafman's research on the largest orchid genus in North America, Platanthera, with several species native to the upper Midwest. Based on the family tree for this group that Jeff calculated from DNA variation and the pollinators of present-day species, he inferred that the ancestral Platanthera were pollinated by nocturnal settling moths, and that from this ancestral condition evolved pollination by day-flying flies, bees and butterflies, by day and night-flying hovering moths, hawk moths, and mosquitoes, and in some cases, selfing arose with a loss of pollinators. The second point is that Darwin did not believe and did not follow up Sprengel's claim that some orchids use deceit to attract pollinators. Today, we estimate that 1/3 of all orchids use deceit, not rewards, to attract pollinators. One example is our native pink lady slipper. The labellum is lobed and conspicuously veined. Bees attracted by that pattern and a nectar-like scent land on the labellum and push their way through the veined opening to access the nectar, and then find that there is none inside, just slick, waxy walls and no way out. But in time, almost every bee finds and climbs up a line of hairs in the back of the pouch and then squeezes under the stigma, resulting in cross-pollination if the bee is carrying pollen from another plant, and then squeezes under the anther, getting brushed with pollen. And it could take that pollen to another lady slipper flower if it makes the same mistake again. Now, orchids use three kinds of deceit involving apparent promises of food, oviposition sites, or mating partners. The pink lady slipper provides an excellent example of generalized food deception. Oviposition site deception occurs in several groups, including the genus Dracula, in which many species bear blood red or black petals and a lip that looks like a gilled mushroom. Dracula flowers emit a mushroom-like scent that attracts fruit flies that lay their eggs on mushrooms, and indeed often lay eggs on Dracula flowers. A recent study using 3D print. Sorry, where did the sound drop out, guys?
Just like the last two.
Or
Okay, well,
half a
let me
sentence.
Okay, so let me just say here that ovocyte deception occurs in several groups, including the genus Dracula, in which many species bear blood red or black petals and a lip that looks like a gilled mushrooms. Dracula flowers emit a mushroom-like scent that attracts fruit flies that lay their eggs on mushrooms, and indeed often lay their eggs on Dracula flowers. A recent study used 3D printing of artificial flowers, combined or not, with scents produced by real flowers to show that both scent and sight cues are needed to attract fruit flies. One of the first convincing cases of mate deceit pollination or sexual deception involved bee orchids of the genus Ophrys, often found in chalk grasslands in Europe. Each species has a labellum that visually mimics females of a local bee species. The flowers emerge before the female bees, and emit the same chemical they do to attract male bees. Because the female bees mate but once, male bees are strongly selected to emerge before they do. And also then to aggressively court and copulate with any females they manage to happen on, often not noticing they're actually not female bees, but orchids. Okay, so what does all this mean for orchid diversity? Based on the phenomena I've just reviewed, biologists have proposed over the years that the evolution of pollinia, or specialization on individual pollinators, or pollination via deceit, or euglossine bees or Lepidoptera, or epiphytism per se, or associated traits like CAM photosynthesis, or distribution in the tropics, and especially in extensive cordilleras, could all have been drivers of the extraordinary diversity of orchids. However, tests of these and other evolutionary hypotheses, including possible patterns of geographic spread, have long been stymied by the absence of a fully resolved backbone phylogeny for the orchids. Resolving relationships within subfamily Epidendroideae, including 80% of orchid species, has proven particularly problematic, as has been resolving the position of the subfamily Vanilloideae with strong statistical support. Based on floral morphology and DNA sequence data, the division of the orchids into 5 subfamilies is now widely accepted. Subfamily Apostasioideae includes only 2 genera and 15 species, all of them terrestrial in tropical forests in Australia and Southeast Asia. Vanilloideae has 180 species, which are mostly terrestrial and are found throughout the Old and New World tropics, with a few temperate species. The lady slippers, Cypripedioideae, have roughly 170 terrestrial species in tropical and temperate regions of Eurasia and the Americas. Orchidoideae is a much larger subfamily with roughly 5,800 species, most of which are terrestrial, found around the world, but most diverse in temperate latitudes. And finally, Epidendroideae is by far the largest subfamily. Epidendroids include roughly 80% of all orchids, and most of those are tropical epiphytes. Only orchidoids and epidendroids have evolved discrete pollinia. Now, to clarify orchid phylogeny and test theories about the impact of various traits on diversification, my colleagues, some of which are online listening to this talk, and I amassed DNA sequence data for 75 plastid genes, much of the chloroplast genome, from 39 species representing all orchid subfamilies and 16 of 22 tribes, as well as 100 outgroup taxa that we used for figuring out what the origin, what the root of the orchid tree was. We used maximum likelihood as a procedure to analyze these data and to reconstruct a fully resolved orchid family tree or phylogeny, with 2/3 of the nodes having very strong bootstrap support, greater than 97%. This plastome tree clarifies broad-scale relationships within the family at several levels and resolved relationships among almost all tribes of the epidendroids. The branching pattern indicates patterns of relationships with the species or groups most closely related being represented by 2 branches that emerge from a deeper branch further back in time to the left. These deeper branches represent inferred ancestors that then spawned a series of descendants, including the species we chose as placeholders to represent the different subfamilies or tribes or subtribes. These hierarchical categories are indicated by the series of nested brackets on the right of this figure, with the subfamilies also indicated by different colors. I should mention, by the way, that the inset family tree to the left indicates the length in terms of nucleotide substitutions of the various branches that we derive immediately from the DNA sequence data. And the 3 branches that have rather great lengths compared with the others represent species that are mycoheterotrophs that have given up photosynthesis, and therefore selection on a lot of genes in the chloroplast genome has been relaxed. And so you can see indeed that's happened, that you have substantially longer branches in those groups. Now note that this phylogeny places the two large epidendroid tribes, Vandae and Cymbidae, at the uppermost part of this figure, sister to each other. Hold on just a second here, folks. I'm having a little bit of difficulty getting there we go, the pointer going here. There are two other large species groups, the tribe Epidendriae and the subtribe Dendrobiinae, among a group of clades or lineages that are successively sister to that recently derived core group of Vandae plus Cymbidae. Several smaller tribes, the so-called lower epidendroids at the bottom of the Epidendroideae in this figure, are successively sister to the rest of the epidendroids. So as you can see, Neodae sister to all other epidendroids, and then Triphoria is sister to the remainder, and then Sobralia and Tripidea and Nervilia and Arethusae are sister to, again, progressively smaller remnants. Sorry. Let me just go back here for a second. Having a little bit of difficulty with the interface, folks. Okay, now to produce a more comprehensive phylogeny with representatives of more groups, we added 144 orchid species represented by sequences for only 3 plastid genes. This is a data set that Ken Cameron contributed to the effort. This allowed us to reconstruct relationships among nearly 200 orchid species, representing all tribes and almost all subtribes. Difficult to resolve relationships involving short branches far back in time were resolved for the first time by our, with statistical confidence by our sequences for 75 chloroplast genes, while more recent, easier to resolve relationships at shallow depths in the tree could be resolved for many additional species just using 3 genes. Now we calibrated the resulting broader orchid phylogeny against time by using the computer program BEAST and 17 angiosperm fossils and their ages. The result, which is summarized on the right, is a fully resolved, time-calibrated tree for the orchids, and a major advance on the previous consensus view of orchid phylogeny. The blue bars indicate the standard deviation of age estimates, and the timescale is shown at the bottom of the tree. Now, based on this study, orchids appear to have diverged from the common ancestor of other asparagales 112 million years ago. And extant lineages of orchids began to diverge from each other about 90 million years ago. The 8 tribes and the 3 previously unplaced subtribes of upper epidendroids diverged very rapidly from each other between 38 and 31 million years ago. We used our time-calibrated family tree of the orchids to estimate rates of species diversification. Here, the heights of the triangles for each tribe or subtribe are proportional to the estimated number of species in each of these groups. Of course, the relationships we've derived from our phylogenetic study. The colors of the branches and the triangles indicate the inferred rates of net species diversification. That is to say, the difference between the rate of speciation and the rate of extinction. And it's calculated using a Bayesian analysis implemented in the computer program BAM. Now, based on this analysis, as shown here, we identified 3 significant accelerations of the net rate of species diversification within the orchids. The first, shown in light blue, at the base of the orchid, pardon me, The first shown in light blue at the base of the orchidoids, and then second in green at the base of the upper epidendroids, and the last in red in the pleurothalids and their allies in the Andes. Warmer colors indicate higher rates of net species diversification. We then overlaid traits thought to influence diversification on our tree and used the computer program BISI to assess whether there were significant correlations of those traits with rates of speciation or extinction or net species diversification. Now, the evolution of Paullinia significantly increased the inferred rates of speciation and extinction and resulted in a 5.1% advantage in net diversification per million years within lineages. Now, a 5% advantage doesn't seem like much, especially if it occurs over 5 million years, over a million years, but it is significant statistically, and it compounds over every additional million years, yielding, for example, an advantage of over 60% in 10 million years. We propose that Paullinia should accelerate speciation by facilitating specialization on individual pollinators or parts thereof, and by reducing the number of independent matings per population. Paullinia may also allow small numbers of variants to produce numerous offspring, promoting speciation from small numerical bases that might otherwise lead to demographic collapse and extinction. Now, epiphytism significantly accelerated both speciation and extinction rates relative to the terrestrial habit, yielding an 8.8% advantage in net diversification per million years within lineages. Epiphytism is a key innovation that allows invasion of a new adaptive zone largely unoccupied by other vascular plants. Epiphytism should also help generate and maintain high diversity because it's associated with growth in highly dissected montane habitats, and because variation in sunlight and humidity within tree crowns permits local niche partitioning by species, and because altitudinal and topographic variation across mountainsides, can be partitioned at broader scales. Now, tropical distributions appeared to accelerate net diversification slightly, but this difference appears driven primarily by growth form, not latitude per se. We found that terrestrial lineages in and outside the tropics had essentially identical diversification rates, while tropical epiphytic lineages diversified 2.3 times faster than either. CAM photosynthesis, the idea of opening up the stomata at night to take up carbon, as you see in cacti and other succulents, and is found in many epiphytic orchids, accelerated speciation and extinction rates, although not significantly, believe it or not. But it did yield a very large and highly significant advantage in net diversification within lineages, and a smaller disadvantage across lineages. Deceit pollination via mimicry of food sources, nesting sites, or potential mates is nearly, not quite, restricted to orchids. It significantly increased speciation and extinction rates, but we found no significant overall change in the rate of net diversification. This remains, I have to say, a puzzle to us. Pollination by male euglossine bees had no significant impact on speciation or extinction rates, but did significantly increase net diversification by 3.6% overall. Euglossine pollination should accelerate speciation by allowing small chemical changes to attract different bee species. Pollination by Lepidoptera doubled speciation and extinction rates and resulted in a 7.1 acceleration of net diversification per million years. Pollination by moths or butterflies involves nectar presentation in corolla tubes or spurs, whose length might rapidly evolve and lead to rapid recruitment or evolution of new pollinators, enhancing diversity. And then finally, life in tropical extensive tropical cordilleras, specifically the Andes and New Guinea highlands whose floras we analyzed, significantly increased speciation and extinction rates, yielding a very large 25% advantage in net diversification rate, presumably because of the large numbers of natural barriers to dispersal of orchids and their pollinators in such areas in the form of ridges and valleys and different climatic zones, promoting speciation in orchids and perhaps in their pollinators, and possibly allowing speciation to proceed in parallel at several points along a mountain range. Extensive tropical cordilleras thus appear to have the strongest overall effect on net diversification in orchids. Life in cloud forests in such cordilleras is, of course, also correlated with acquisition of the epiphytic habit and CAM photosynthesis. Our current statistical approaches do not allow us yet to separate out the partial effects of those correlations on the drivers of orchid diversity. Okay, then next here, if I can get the screen to advance. Little challenging here. Sorry for a second, folks. There we go. Okay, so we've shown that orchids are remarkably species rich, partly as a result of 3 accelerations of net diversification. Our data provide the first quantitative support for several hypotheses regarding the genesis of orchid diversity and identify specific points in orchid evolution where these factors play a role. They show that multiple factors, several that interconnected, have contributed to orchid diversification. The first significant acceleration of net diversification is at the base of the orchidoideae, as I've indicated here with the blue box, just one node after Paullinia evolved. The second acceleration coincides with the rise, with the origin of epiphytism in the upper epidendroids and the rise of several montane clades. A significant deceleration shown by the gold box within the upper epidendroids reflects the rise of a grade composed of temperate terrestrial orchids and lowland epiphytes in the tropics. And the final acceleration in tribe Epidendriae coincides with a large concentration of Andean taxid pollinated, often by deceit or Lepidoptera. Florothalids, the most diverse part of this clade, are largely pollinated by flies lured by deceit. Low diversification rates characterize the lower epidendroids, which are terrestrial and mainly lack pollination via deceit euglossians or Lepidoptera. Overall, our results show that epiphytism and life in extensive tropical cordilleras, as well as pollination by Lepidoptera and euglossian bees, have had large effects on orchid diversification. Interestingly, the defining characteristics of orchids, tiny seeds, mycoheterotrophic germination, and the floral column, did not accelerate diversification by themselves. That is to say, the rate of diversification in orchids, looking at the ancestral orchids, did not differ from other asparagales, but we suspect, and we have specific scenarios by which they may have facilitated subsequent diversification after Paullinia and epiphytism evolved. Okay, biogeography also matters. We used our family tree and the distribution of present-day species to reconstruct the distribution of ancestors, represented here by pie diagrams deep in the tree, with a probability of origins in continental regions indicated on the map, signified by the width of slices with the same colors in the pie diagrams. Our biogeographic reconstruction implies that orchids arose on the Australian plate 112 million years ago. By 90 million years ago, they had dispersed to South America at a time when Australia, Antarctica, and South America were in close contact, and Antarctica had a subtropical climate due to a much greater concentration of CO2 in the atmosphere then as opposed to today. We infer that orchids dispersed, therefore, over land from the Australian plate to South America via Antarctica. Thus, while Antarctica is the one continent where orchids do not now occur, it appears that Antarctica played a key role in their early evolution. Despite their tiny dust-like seeds, orchid appear to have successfully undergone long-distance dispersal across oceans and between biogeographic regions less than once every million years on average. 97% of orchid species are indeed restricted to these individual biogeographic regions. And that suggests that slow geographic spread and speciation in situ has played a major role in orchid diversification, with lineages diversifying in parallel as a result of that limited dispersal on several continents at the same time. Let's see if we can get the next slide growing here. There we go. Now, over the last 48 million years, and don't worry about all those numbers, within the epidendroids, which contain over 80% of all orchids, the highest rate of diversification occurred in the neotropics and on the Australian plate, including New Guinea, and indicated that highly significant difference there, and the effect of those areas on diversification rates are respectively 11.5% and 9.1% per million years. So a very big effect of both of those areas. And in addition, the neotropics served as the most important of all of the source areas for orchids in other regions. That's a seeding in new lineages to other regions. And the tectonically active and topographically complex region of Southeast Asia served as the most important target area, receiving colonists from several different areas. But even this is not the whole story. Several factors implicated in orchid diversification also appear based on other studies by my lab group to generate the high diversity of bromeliads. Yet bromeliads have evolved fewer than 3,300 species. Why do orchids have nearly 8 times as many species as bromeliads? Now, 4 factors might help account for this difference. First, dispersal, time, and familial range. The dust-like seeds of orchids, the greater cold tolerance, and probably most importantly, the much greater time since the beginning of their initial diversification, 90 million years ago in the orchids versus 23 million years ago for bromeliads, almost surely led to greater orchid diversity by permitting them to spread to every continent and most latitudes, while bromeliads remain confined to tropical and subtropical America and have only been diversifying for about a quarter of the time. Secondly, chromosomal evolution. It's often overlooked in orchids, and yet many of the largest genera show extensive variation in chromosome number. Of course, the question is always chicken and egg, which came first. Pollinia and tiny seeds, as I've mentioned before, allow recovery from repeated population bottlenecks in orchids. Such bottlenecks could fix karyotypic mutants via drift, quickly generating post-mating barriers, preventing hybridization between species with different chromosome numbers, and as a result lead to rapid speciation. Third, limited dispersal of pollinators. Again, I'm sorry, I'm getting problems here in terms of getting the slides to advance. 1,000 pardons. Right, so limited dispersal of pollinators might be involved, or limited physiological tolerance of montane pollinators might be involved. The evolution of dozens of closely related species of Tegia over only a few kilometers and Andean Ecuador, as documented by Lou Jost, and more generally the very rapid geographic turnover of epiphytic species with distance in the Andes are both consistent with gene flow over only short distances in tropical montane areas. More research is needed to determine whether this might reflect the limited dispersal ability of the small, soft-bodied, desiccation intolerant flies that pollinate so many individual species of orchids there. Finally, greater species packing. Across 17 neotropical forests surveyed by Kreft, epiphytic orchids had nearly 6 times as many species per stand as bromeliads, and more geographic turnover from site to site. Higher levels of local diversity suggest that community assembly in orchids may involve a finer or more extensive partitioning of pollinators or perches than in bromeliads. Now, to the ecological and evolutionary factors affecting orchid diversity at global or continental scales that I've discussed today must be added factors determining diversity at smaller scales within continents or local landscapes. Some of these factors involve quantitative variation in factors I've already mentioned, like how much area and what elevational range do particular tropical cordilleras occupy. We expect in general that areas with a greater range of environmental conditions, like a greater range in elevation, rainfall, temperature, and soil fertility, should support more species. So should areas with greater topographic dissection. They ought to support a greater diversity of species adapted to different conditions and provide natural ecological barriers that encourage speciation in situ. Mid-elevation habitats that favor cloud forests favor the species-rich epiphytic guild and therefore favor more species. Nature reserves should target areas with more native species, and especially narrowly distributed native species which are found in few, if any, other places, and thus would add to the list of species protected and would include species that are inherently at high risk. Now, as you can see from the map and the graph, among continents and subcontinental areas, South America is exceptionally diverse given its size, almost surely due to the presence of the tall and recently uplifted equatorial Andes, with heavy rain and fog swept in over the Amazon, and the high diversity of pleurothalids and other highly diverse epidendroid groups in the cloud forest there. At the same time, topographically dissected and or climatically diverse smaller areas support proportionally more orchid species in Malaysia, Central America, New Guinea, Southeast Asia, Mexico, Southwest China, Madagascar, and Southwest Australia. In part, the lower ratio of species to area in South America simply reflects species area curves. The number of species expected, other things being equal, should increase with only the 1/5 or 1/4 power of area. But South America far outperforms the other continents in orchid diversity, with most having a sixth or less of the species found in South America. Asia, with an area twice that of South America, has 2,000 fewer species. Now you can see that Australia also has a rather low number of species, though it has more than North America if you exclude Mexico. Let me just see if I can get the slides again to advance. Having a problem. But it turns out, as I'll get to the next slide, if I can ever get the slides to advance, that Australia orchids are mainly restricted to the green rim, to the mesic habitats near the coast. Boy, I'm going to have to maybe, I don't know what I'm going to do here. I can't get it to advanced.
Try now.
Okay, thanks. And because of this concentration on the green rim, where more mesic conditions are found, Australia supports a surprisingly large number of species for the area involved. Finally, at smaller scales, gap analyses and diversity modeling in Central and South America have repeatedly shown the importance of dissected mid-elevation montane regions as orchid hotspots, meaning hotspots for epiphytic orchids. In Bolivia and Peru, Kessler and his colleagues have shown high local species diversity, and epiphytes generally peak at mid-elevation cloud forests, and epiphytes show high compositional turnover in moving across the landscape at a given elevation. species that are narrowly endemic to particular areas help create that pattern and put a premium on putting preserves in such areas. I would add that preserving montane gradients that cover a substantial range of elevations would have the additional advantage of buffering against habitat losses due to climate change, allowing species simply to move uphill over short distances as conditions warm. Such gradient preserves should thus be a priority, especially in areas of the highest plant diversity, including the Andean slopes above northwestern Amazonian, the rainiest part of the Amazon basin. I expect the next two speakers in today's symposium will address many of these issues, so I'll end here and take questions, though I would like to finally express my deep gratitude to National Science Foundation for its continuous support since 1987, and to my colleagues for their many contributions to the study of monocot evolution, bromeliad evolution, and especially in today's context, the investigation of orchid evolution and diversification. Thank you so much.
Thank you, Tom. We have a number of questions in the chat box, and Ron also suggests if you have questions, you can raise your hand. To raise your hand, you hit alternate Y if you have a Windows machine, or option Y if you have a Mac. So the first question I have from Ashley Gable, she wanted to understand how CAM photosynthesis affects diversification.
Yes, most likely it affects diversification by allowing epiphytes to grow despite their lack of contact with soil moisture. So in most species that have so-called C3 photosynthesis, the stomata opens, CO2 diffuses into the leaf, and the chloroplasts absorb the CO2 and they fix it as sugars using energy supplied by sunlight. The problem is the stomata have to be open to let the CO2 diffuse in, and that means water diffuses out. And that is a problem for plants that have access to reliable supplies of moisture. And so some plants have evolved another mechanism of photosynthesis to cope with water shortages. This is called CAM photosynthesis. And in this case, what happens to try to reduce water loss, the plants do not open their stomata by day, they do it by night. And they will absorb CO2 and store it as an organic acid. They're not actually, they don't have energy, right, to reduce, chemically reduce carbon dioxide, this oxidized forms of carbon, to an unoxidized, energy-rich form of carbon, like sugars. But they store it as an organic acid. And then they close their stomata, and then by day, when the sun comes up, energy comes in, they release carbon dioxide from the organic acids and take up CO2 without losing water by day. It greatly reduces the amount of water loss because it's much more humid at night. But it also greatly reduces the total amount of photosynthesis that could be done because it's limited by how much malate, how much organic acid can be stored. And so a number of groups have evolved CAM photosynthesis in drier areas, especially in, it turns out, epiphytes in tropical forests. Now, the forests are often quite wet, but the branches aren't, right? They dry out really fast. And so that appears to be why orchids, and indeed many bromeliads, have evolved CAM and allowed them to become epiphytic. And then by being epiphytic, there are a number of advantages that lead to epiphytes speciating rapidly. Does that answer your question?
Yes, thank you very much.
Very good, my
Okay.
pleasure. Okay.
Then
questions?
Ron Parsons had several questions, so rather than me trying to interpret them, I'm turning on his mic and he asked you directly. So
Okay.
Ron?
Hi, Ron.
Hi, Tom. How are you?
Good, thanks.
Hey, listen, I was looking at the charts and stuff a lot while you had them up, and I was just kind of curious. If there are many genera like the Cephalanthera. You got the Austinii, which is microheterotrophic, but then you've got photosynthetic species with the, so are there many genera like Ed and Neodia that have both species, both types of?
There certainly are a handful, and one of the big questions, of course, is how widespread maybe the phenomena of partial mycoheterotrophy might be. Now, all orchids are mycoheterotrophic, right, initially, to produce the protocorms. but then they usually give up and photosynthesize. But recent work in Europe, for example, suggested that a number of orchids are partially mycoheterotrophic as adults. They're still green, right? Unlike, say, the Cephalanthera that you're mentioning, that's white and clearly is not photosynthetic. And one of Ken Cameron's students, Brendan, Corder, is pursuing just that issue now, and actually he's found a number of species in our flora that are partial microheterotrophs. As far as the exact question you asked, there are many cases of that mingle photosynthetic and mycoheterotrophic species. There are a few, you've mentioned a few of them, but another one that's recently been added to the list is Platanthera. And there will be a paper appearing shortly in Nature Plants that analyzes for the first time the genetic changes in the nuclear genome that have accompanied the transition from fully photosynthetic to partially mycoheterotrophic to fully mycoheterotrophic. For the last decade, people have been poring over the easily sequenced, relatively easy sequenced now, chloroplast genome, but this is the first time that comparisons have been possible. Does that answer that question?
Yes, yes. And then the other ones, just real briefly, on the chart you had, in one chart you had Angricinae included in the Aridinae. And I was wondering if that was a recent development, but then on another chart you had Aranginae, Angricinae, Aridinae, and I think the Polystachyonae were all in the, yeah. So I was wondering if so I understood, excuse me, I understood that the Aranginae was put into the Angricinae, and that was now the new group for all of the angricoid orchids of Africa and Madagascar. But I was wondering, because the charts are what confused me. What is the current status of those?
So keep in mind that these charts were published in 2016.
Okay.
And there have subsequently been some name changes. So I'm going to have to, without getting into the weeds here, leave it at that. So, but I will add to this that, this was just a preliminary survey, and what needs to be done is basically at least sequence plastomes across large numbers of orchids to increase the resolution of the tree. That, in fact, has been done, is increasingly being done. I mean, the paper is coming out every year on this now.
Sure.
And what also needs to be done, and has been done, is to replicate the study using nuclear genes. And that has been done. That's appeared in the last year or two. And that largely confirmed our perspective in terms of the relationships among different tribes and subtribes, with a few changes, of course, but otherwise largely the same story.
Okay. And one last question: are you coming out for Calicornis this year?
Man, I wish I would. I've killed 3 weeks of my life writing a $2.5 million proposal to NSF, and getting a little sick in the process. So I canceled my trip to California to see Calicornis, and I'm going to. I was going to do it last week, and I'm hoping to come sometime in late April. I don't know. Will they all be gone by now, though? By then, though?
No, not at all. May and June is the best month.
Well, then
Yeah.
I may do that, because I'm taking my students to the Smokies and the Southern Appalachians in 2 weeks, so I can't go then, but I can do it after classes end.
We have a Q student coming at the end of May for a week, for May, end of May, early June. So there's plenty of stuff then.
Oh, very good. All right.
Okay,
Well,
now-
I'd be delighted to do that. I'll take you up on that offer. Good to
Thank you,
see
Tom.
you and Mary again.
Thank you.
Okay, that'd be great. Thanks, Tom.
Thanks.
One last question, and then I think we have to move on.
Sure.
Mark Wilson asked, do you have any thoughts on the significance of polyploidy in speciation?
So yes, I think that it's been demonstrated that polyploidy leads directly to speciation, right? Because of incompatibilities during sexual reproduction. Triploids lead, for example, to inability to successfully complete meiosis. And the Sultai, Doug and Pam Soltis, have argued that polyploidy has been a big impetus for speciation at a different level in terms of duplicating the genome and allowing perhaps specialization of different copies of the same gene for different purposes. I have not analyzed the distribution of ploidy across Orchidaceae to relate it to the diversification rates. That would be a very exciting thing to do, but we haven't done it. The only thing, as I mentioned, was that it is interesting that many of the largest genera have a lot of different chromosome numbers, and I suspect that there may be a causal relationship there from going from changes in chromosome number to more speciation. Okay.
Okay, thank you, Tom. There's a few more questions, but what I've asked people to do is to directly send the questions to you, Tom. That's okay, because we ran out of time. And I'm going to turn it over to Ron now for whatever you have planned next.
Great. Thank you very much, Tom. Thank you, Mary. Excellent kickoff. So we're running a few minutes late, but that's okay. We're not on a strict time clock. So why don't we go ahead and take a break for about 5 minutes, and people can do whatever they need to do, and we'll reconvene and get on to our next speakers.